Hello students, in today's lecture we are going to discuss MCQs and PYQs on prokaryotic cell. First MCQ, the cell envelope of prokaryotes consists of A option glycocalyx, B cell wall, C cell membrane, D all of these. So answer is all of these. D. Next question, many ribosomes may associate with a single messenger RNA molecule to form multiple copies of a polypeptide simultaneously. Such chain of ribosomes are termed as. So this question was asked in NEET 2018. So option A peroxisome, option B plastidome, option C polysome and D polyhedral chain. So our answer is polysome. जब बहुत सारे राइबोसोम्स मैसेंजर आरएनए मॉलिक्यूल को अटैच होते हैं तब जो चेन तैयार होती है साइटोप्लाज्म में उस चेन को कहते हैं पॉलीराइबोसोम और इट इज़ ऑल्सो कॉल्ड एज ए पॉलीसोम एंड दैट्स व्हाई आवर आंसर इज पॉलीसोम. अभी यहाँ पे पॉलीपेप्टाइड चेन क्या है? पॉलीपेप्टाइड चेन बोले तो प्रोटीन मॉलिक्यूल. एक प्रोटीन मॉलिक्यूल में पॉलीपेप्टाइड चेन हो कितने भी हो सकते हैं. कोई प्रोटीन मॉलिक्यूल में एक होती है, कोई प्रोटीन मॉलिक्यूल में दो पॉलीपेप्टाइड चेन होते हैं. तो प्रोटीन्स आर मेड अप ऑफ पॉलीपेप्टाइड चेन्स और राइबोसोम्स आर साइट ऑफ प्रोटीन सिंथेसिस. कि जब मेनी राइबोसोम्स अटैच होंगे सिंगल मैसेंजर आरएनए मॉलिक्यूल को तब एक राइबोसोम में एक प्रोटीन मॉलिक्यूल तैयार होता है. तो मेनी राइबोसोम्स जब अटैच होंगे मैसेंजर आरएनए मॉलिक्यूल को तो साइमल्टेनियसली मल्टीपल कॉपीज ऑफ पॉलीपेप्टाइड चेन्स are formed in the cytoplasm of bacterial cell. Now next question, which of the following components provide sticky character to the bacterial cell? So this question was asked in 2017 Delhi CBSE exam. Option A cell wall, option B nuclear membrane, option C plasma membrane, option D glycocalyx. So here answer is D glycocalyx. So we know that bacterial cell envelope consists of three layers. First outermost is the glycocalyx, second is the cell wall and last one innermost is the plasma membrane. So this glycocalyx in bacteria is made up of capsule or either slime layer, and this capsule and slime layer they gives the stickiness to the bacterial cell, and so that bacteria can attach to the rocks in water surfaces. And pathogenic bacteria, if they have capsule, they can easily attach to the host tissue and therefore they can invade or they can cause the disease in host tissue. Now coming to fourth question, the genetic material of prokaryotic cells is called A option nucleus, B nucleolus, C nucleoid and D centrosome. So answer is nucleoid. So we know that bacteria do not have membrane bound nucleus. Its DNA is necked, and that necked DNA present in the cytoplasm of the bacterial cell is called as a nucleoid. It is also called as a bacterial chromosome. Next question, cell wall of prokaryotes is made up of A option chitin, B cellulose, C glucosamine, D mucopeptide. So our answer is mucopeptide. So what is mucopeptide? Mucopeptide is a complex molecule which is form of carbohydrate and proteins, and such complex molecule it gives the strong support to the bacterial cell because cell wall of bacteria is made up of mucopeptide which is a strong rigid molecule. That's why our answer is mucopeptide. Next question, which of the following is not a function of mesosomes? A option respiration, B is DNA replication, C increases enzymatic content and D reproduction. We know that mesosomes are nothing but the membranous structure which is formed due to the extension and folding of plasma membrane. तो mesosomes का function क्या क्या है? It helps in respiration process because in aerobic bacteria when mesosomes are formed they increases their respiration rate. They help in respiration process. And when cell divides at that time DNA replication first takes place and DNA molecules get distributed into two daughter cells. At that time mesosomes help to distribute the DNA molecule after replication, and it also increases the enzymatic content due to the increase in the membrane structure in the cytoplasm of the bacterial cell. And but last option, our answer is D. Why D? Because mesosomes do not play any role in reproduction and therefore answer is D. Next question, larger subunit of prokaryotic ribosome is A option 30S, B option 40s, C option 50s, and D option 60s. We know that ribosomes in prokaryotes are made up of two subunits. Larger subunit is a 50s subunit and smaller subunit is a 30s subunit. And these two subunits together when present form the 70s ribosome in prokaryotes. So the larger subunit is 50s and smaller one is the 30s. So that's why the answer is C. Next question, gas vacuoles are not reported in option A cyanobacteria, option B blue-green photosynthetic bacteria, option C amoeba, option D prokaryotes. We know that in amoeba gas vacuoles are absent because amoeba is eukaryotes, and in some prokaryotes gas vacuoles are present such as cyanobacteria and purple green photosynthetic bacteria. They possesses gas vacuoles in the cytoplasm of their cell, and that's why answer is C. Amoeba does not possesses gas vacuoles. Now next question, prokaryotic cells does not have A option nucleolus, B membrane bound organelles, C centrioles, all of these. So answer is D, all of these. So in prokaryotes membrane bound organelles are absent, true nucleus is absent, therefore nucleolus is also absent. Centrioles are present in animal cells which are eukaryotes, and therefore the answer is all of these. Next question, in prokaryotes the chromatophores are A option a specialized granules responsible for coloration of cells, structures responsible for organizing the shape of the organism, C inclusion bodies lying free inside the cells for carrying out various metabolic activities, and D option internal membrane system which becomes extensive and complex in photosynthetic bacteria. So here the answer is D, because these chromatophores are present in photosynthetic bacteria, and chromatophores are formed due to the extension and folding of the प्लाज्मा मेम्ब्रेन. तो ये क्रोमेटोफोर्स क्या है, मेम्ब्रेन सिस्टम है जो प्लाज्मा मेम्ब्रेन के एक्सटेंशन से और फोल्डिंग से फोटोसिंथेटिक बैक्टीरिया में तैयार होती है, और उसमें पिगमेंट्स होते हैं जो फोटोसिंथेसिस प्रोसेस के लिए बैक्टीरिया को हेल्प करते हैं. Now 11th question, which layer of the cell envelope determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing? A option cell wall, B cell membrane, C glycocalyx, and D capsule. So here answer is A. So we know that cell wall is the second layer of the envelope in prokaryotes. Below capsule there is a cell wall, and this cell wall, it is made up of mucopeptide, a complex chemical compound, and therefore this cell wall, it provides a strong structural support to the bacterial cell. Thereby the bacterial cell do not get burst or collapsed due to the external environmental factors and environmental pressures. So cell wall क्या है, एक लेयर है सेल एनवेलप की जो बैक्टीरियल सेल को स्ट्रॉन्ग सपोर्ट प्रोवाइड करती है, क्योंकि ये जो सेल वॉल है ये कॉम्प्लेक्स केमिकल कंपाउंड से बनी हुई होती है, एंड देयरफॉर इट प्रिवेंट द बर्स्टिंग ऑफ बैक्टीरिया, कि वो बैक्टीरिया को कोई भी एनवायरमेंटल प्रेशर हो, एटमॉस्फेरिक प्रेशर हो, उनके फैक्टर से बर्स्ट नहीं होने देते, कोलैप्स नहीं होने देते. नेक्स्ट क्वेश्चन, मेसोसोम्स आर द इनफोल्डिंग्स ऑफ सेल मेम्ब्रेन. दे A option help in cell wall formation, DNA replication and respiration. B option increases the surface area of plasma membrane. C option are present in both prokaryotic and eukaryotic cells. Choose the correct option. Out of these three options we have to choose correct option. तो यहाँ पे पहला ऑप्शन क्या दिया है, मेसोसोम्स हेल्प इन सेल वॉल फॉर्मेशन, डीएनए रेप्लिकेशन एंड रेस्पिरेशन. सो दिस ऑप्शन इज करेक्ट. Increases the surface area of plasma membrane. This is also correct. Which is incorrect then? Are present in both prokaryotic and eukaryotic cells. तो ये जो ऑप्शन है ये इनकरेक्ट है, क्योंकि मेसोसोम्स प्रोकैरियोट्स और कुछ प्रोकैरियोट्स में ही प्रेजेंट रहते हैं, यूकैरियोटिक सेल में एब्सेंट रहते हैं. दैट्स व्हाई सी ऑप्शन इज रॉन्ग. सो करेक्ट ऑप्शन इज देन सी. Mesosomes are not present in prokaryotes and eukaryotes. Therefore, answer is C. Option A and B are correct. Option C is incorrect. Next question, which of the following statements about inclusion body is incorrect? This question was asked in NEET 2020. Option A, these are involved in ingestion of food particles. Option B, they lie free in cytoplasm. Option C, these represent reservoir material in cytoplasm. Option D, they are not bound by any membrane. So here answer is A. So what are inclusion bodies? They are non-membrane bound reservoir food material present freely in the cytoplasm. तो inclusion bodies क्या है? Non-membrane bound है, साइटोप्लाज्म में फ्रीली दिखाई देते हैं और वो क्या करते हैं? रिजर्व फूड सब्सटेंसेस, एक्स्ट्रा न्यूट्रिएंट्स स्टोर करके रखते हैं. दैट्स वाई इनकरेक्ट ऑप्शन इज ए. दीज आर इन्वॉल्व इन इंजेक्शन ऑफ फूड पार्टिकल्स, कि वो फूड पार्टिकल्स का इंजेक्शन नहीं करते हैं. दैट्स वाई आंसर इज ए. नाउ 14th क्वेश्चन. बैक्टीरिया शो ए रेंज इन द नंबर ऑफ अरेंजमेंट ऑफ फ्लैजिला. बैक्टीरियल फ्लैजिलम इज कंपोज्ड ऑफ ए ऑप्शन 2 parts pila and fimbriae, B option 3 parts filament, hook and basement membrane, C option 3 parts filament, shaft and basal body, D option 3 parts filament, hook and basal body. So answer is D. So bacterial flagellum consists of 3 parts: filament, hook and basal body.